Hi students, it's Mrs. Sanzone. I'm here to do another riveting uh, video note for you on discovering cells. I haven't done this in a while, so I apologize. Make sure you have a piece of Cornell notepaper in front of you or that you've printed out a copy of the skeletal notes that I made available on Google Classroom. And our title today is Microscopes in Cell Theory. This is what we read about in class yesterday, and some of you may have had to finish it for homework last night. We do have an essential question today, and the essential question is how did the invention of the microscope contribute to knowledge of living things? So once the microscope came about and we were able to see things a lot closer, how did that change our idea of what living things were made of and what living things did? Um, please note, oops, I spelled book wrong. That's good. Please note that we are working in a new book now. We're working on chapter 1, section 1 of Discovering Cells, which is in the Cells and Heredity book, which we read from yesterday. Now let's get into our notes section. So the first part of our notes is going to be about an overview of cells. And we're going to start with cells and structure. Um, we did talk quite a bit about what structure meant when we discussed cells at the very, very beginning of the year. We talked about the characteristics of living things and that all living things are composed of one or more tiny cells, and that the definition of a cell was the basic unit of structure and function in living things, which I've told you time and again that I hate that definition because I don't really understand what that means on the surface. So let's talk about structure. What is structure? Structure is just what is something made of? How is it built? So I'm putting a little picture of a building here. Da 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 da, we have a building. Um, Obviously, cells aren't made up of tiny buildings, but they do look something like this. This is an example of an animal cell, which you're going to learn a lot about in the coming weeks. Um, so everything that living things are made of, like how they're made, how they look physically, is because of cells. And that's what cells and structure basically says. Um, so the structure of living things are determined by the amazing variety of ways in which cells are put together. Um, Obviously, the more complicated the organism, um, the more variety you're going to have in those cells. So, for example, if you just have one simple bacterial cell, a bacteria is just one cell. It's not going to be very complicated. But when you're talking about a human's not going to only have brain cells and cells for your eyeballs and cells for your heart and cells for your liver and your stomach and muscle cells and skin cells. All of those cells, they're all animal cells, but they're all different and they're going to function in a variety of ways and they make us who we are. Um, that's what structure is all about. Next, we're going to talk about cells and function. So if cells are the basic structure and function of living things, so they're the basic makeup of what they look like, their physical being, but also their function. So what do living things do? And here I have a picture of my kids. Surprise. Shocker that I'd want to talk about them, I know. So here's a picture of my little goobers and the dog on Halloween. And what are they doing? What's their function at that point? Well, they're dressed up in Halloween costumes. Maya is Hermione, and Ava is Professor Snape, and Lena is Professor McGonagall, obviously. So everything, not only that living things are made up of, but also what they do is because of cells. And so we're going to add that in. Cells are involved in so many different things. And what our book talks about is that cells are involved with obtaining oxygen, getting rid of waste, obtaining food, and growing. There are a lot of other things that cells are involved in, and we're going to get into that a little bit more. But this is a good basic overview of what they do. And also we need to note that cells are so numerous and they're really, 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 really tiny. Most cells you can't see without a microscope. So why didn't people think cells existed when they were talking about living things back in the 1500s? Well, because they didn't know cells existed yet. And that's because they're so small and we needed microscopes with really good lenses in order to see them. Um, most cells are way too small to be seen with the eye. 1 square centimeter of skin contains more than 100,000 cells. Yesterday, we looked at the quark cells under the microscope, and Robert Hooke described 1 square inch of quark having over 12 million cells. Just try to wrap your brain around that a little bit. It's really, really unbelievable. I've actually pulled in a picture here. These are neurons. Neurons are brain cells. And on average, we have about 100 billion, billion with a B, 100 billion brain cells in our brains. And your brains really aren't that big. If you take your two fists and you put them together, that's about the size of your brain. So think about that. Having 100 billion of something within that small, small space is really kind of baffling when you think about it. So when did people first start knowing about cells? Well, the invention of the microscope really made this possible. Obviously, you've seen microscopes before. We used one yesterday in class, but I'm going to pull in a little picture. Here's a pretty, not very complicated one. Yesterday I used one that involved having down here an electric light source for its lamp, but here we have just a mirror. We do have some microscopes like this too. Some of you might end up liking them a lot. But it's the microscope that made it possible for us to discover what cells are. And all microscope is an instrument that makes small objects look larger. Obviously, you can have a microscope, whoops, like the one pictured here, that involves more than one lens, and it has a stage and it has all this stuff on it. And that's definitely a microscope, but even just like a magnifying glass in a way is considered a microscope as well. That's a simple microscope. So how did this all start? Well, these two guys, Hans and Zacharias Janssen, they were eyeglass makers. And they were Dutch, and they made one of the first compound microscopes. So they had a tube with a lens attached at each end, and that allowed them to see things a lot closer up. Obviously, the fact that they were eyeglass makers really, really helped their cause because they had lenses at their disposal. I'm not going to talk too much about those two. Next, we're going to talk about Robert Hooke, because he's the guy who really came up with the name cell. He discovered cells. So in 1663, Robert Hooke used a compound microscope similar to the one that Hans and Zacharias Janssen had used, and he looked at a slice of cork, which is the bark of a tree, and saw these tiny little rectangular rooms, and he decided to call them cells. So here's a picture of Robert Hooke, and here he is. He has his microscope over here. He obviously didn't have electricity available to him at that time, but he did have a lamp, and he used a mirror, and he was able to focus the light, and he was able to see quark cells. So yesterday when you looked under the microscope, you probably saw something quite similar to this, and those are quark cells. After Robert Hooke, the next important person is Anton van Leeuwenhoek, and no, I'm not going to break out into song. And Anton van Leeuwenhoek used a simple microscope. So again, a simple microscope is just one lens, but the lens he happened to use was made what he was looking at 266 times bigger than what it actually was. So it was actually very, very good magnification for just a simple microscope. And he saw little single-celled organisms, and he decided to call them animalcules. So here's a picture of Anton van Leeuwenhoek. And I did find another picture here of Anton van Leeuwenhoek and some animalcules, although I'd like to call this false advertisement because he didn't use a compound microscope to look at animalcules. He actually used something like this. It's just a simple microscope. Um, and this wasn't his job. He wasn't a scientist. He wasn't somebody who did this for a living. He was a guy who was just tinkered around, and he liked playing with lenses and making little microscopes and looking at things. So it's kind of neat that this is somebody who's just had a different profession, but was just fascinated by science, and he ended up making a really important discovery in the process. Okay, next we have Carl Zeiss and Ernst Abbe. And many of you probably have cameras with Zeiss lenses on them, and that's because of this guy. And they made a compound light microscope that made images much clearer, and they used mirrors to focus light through a specimen that made it really, really possible to see it very well. Here's a picture of Carl and Ernst, and here's a picture of the type of microscope they used. And now this was in 1886, I believe, and this really doesn't look very different from those microscopes that I showed you that we have from like the 1960s in our classroom. Believe it or not, it's just really good technology. So just to recap, observation of cells came about because of the microscope. Started with Hans and Zacharias Janssen, who made eyeglasses and made a compound microscope with a tube and two lenses. Robert Hooke used one of these compound microscopes to look at a slice of cork and saw little tiny rectangular rooms in the slice of this plant and decided to call them cells. Then Anton van Leeuwenhoek, using a simple microscope, looked at pond water and water from puddles and scrapings from his teeth, which really grosses me out, and was able to see what he called animalcules, these little things moving around. Then you have Carl Zeiss and Ernst Abbe, who come up with a compound light microscope that really makes images a lot clearer, and it's very similar to what we are going to be using in our class. So based on all this, cell theory was able to develop. And the people who are really responsible for that start with, first of all, so the use of microscopes really facilitated this. Being able to see that cells existed in the first place obviously is going to make it possible to come up with ideas about what cells are and what they do. The first guy, Matthias Schleiden, looked at a lot of plant cells, or a lot of plant specimens on a microscope. And over time, looking at all these plants, he could see that they always looked kind of similar, and they had these little boxes, and he came up with the idea that all plants are made up of cells. And here is an example of a plant cell, and you will learn a lot more about this, and you'll be able to name all those little parts called organelles in a few weeks. So that's Schleiden. And I should put that in there. Hold on. So Schleiden came up with this idea after looking at all these slices of plants. All plants are made up of cells. Then came another guy, Theodor Schwann, who took a lot of time looking at different animal specimens, and he saw something familiar over and over again. And here's an example. This is some blood cell, animal blood cell, and he came up with the idea that, okay, well, all animals are also made out of cells, just like my buddy Schleiden said that all plants are made of cells. I'm going to make a statement here that's, you know what, all animals are made up of cells. And guess what? If all animals are made up of cells, that means, wait a second, all living things are made up of cells. So he kind of was able to jump to that conclusion. So here we do, sorry, Here's a picture of living versus non-living things, and all of these things over here, they're alive. And guess what? If they're alive, thank you, Mr. Schwann, they're made up of cells. These things over here, not so much. Okay, moving on. Now we have Rudolf Virchow. And Rudolf Virchow, he really, so he saw what Schwann and Schleiden had done about plants and all living things and animals being made up of cells. And at the time, there was this whole idea about spontaneous generation, that living things could just erupt from things that weren't alive. And there were a lot of experiments going on, such as Francisco Redi. We talked about that at the very, very beginning of the year, who proved that spontaneous generation did not occur. It's not possible. Living things come from other living things. So from that, Virchow was able to say, okay, well, if all life comes from life and all living things are made up of cells, well, guess what? Then all cells come from cells. Okay? And so then we end up having what we call cell theory. And the cell theory says three really, really important things. Number one, all living things are made up of cells. Oops. That's super important. You knew that already. Number two, cells are the basic structure. Oh, that didn't work. Hold on. Edit. Cells are the basic units of structure and function in living things. That's our fun definition that we've gone over. So everything that a living thing is made up of and everything a living thing does is because of cells. And number three, all cells are produced from other cells. Hooray for cell theory. You were going to know this so well. I'm so excited. Okay, so again, we have Schleiden, Schwann, and Virchow contributing to the idea of cell theory, that all living things are composed of cells. Cells are the basic units of structure and function in living things, and that all cells are produced from other cells. Moving on, let's talk about, briefly, we're going to talk about light and electron microscopes. Now, I'm not going to spend a ton of time on this right now because starting on Wednesday, we're going to really go into detail about microscopes, about what they're made of, and all the different parts and how to use them. And so you're going to know this stuff pretty well. First, let's start with magnification and lenses. So lenses in light microscopes magnify an object by bending the light that passes through them. So you have a couple different kinds of lenses. Convex lenses are really, really thick in the middle and thin at the edges. Concave lenses, well, they cave in, so they're really thick at the edges and thin in the middle. Microscopes use convex lenses because those focus light in and make things look bigger than they really are. So magnification is very, very important because obviously it's going to make things look bigger. And even better than just magnifying something is using more than one magnification. So the microscopes that we're using are compound, meaning they use more than one lens. And so the benefit of that is that you take the magnification of one lens. So let's say you have one that makes something look 10 times bigger than it really is. And then you take the magnification of the other lens. Let's say that also makes something look 10 times bigger than it really is. If you're able to use both those lenses together, that's going to make something look 100 times bigger than it really is. So you get to multiply those two magnifications together. And so you get so much better magnification as a result. So compound microscope magnification uses more than one lens. The two lenses magnifications are multiplied together to get the total magnification. So super important, magnification, making something look bigger. And there you go, you have a creepy eyeball. But of course, making something look bigger is all well and good. But if you don't have a clear image, that's not going to do you any good. So you need resolution as well. So magnification makes something look bigger. And resolution is making it look clear or sharp. So the definition in our book, resolution is the sharpness. Okay. Resolution is the sharpness of an image. So how clear is it? So here's a little graphic here. So obviously here it's not very sharp, but as you move on, obviously the resolution here is a lot better than the resolution here. I think a lot of you are probably more familiar with resolution than previous classes because of like video games have gotten a lot better and televisions and things like that. So as far as microscopes are concerned, magnification and resolution are going to be the most important things. So making it look as big as possible and as clear as possible are super important. Now, our compound light microscopes that we're going to be using in class, they do a pretty good job, but obviously they're not the best of the best. When you're talking about the best of the best, you're really talking about things like electron microscopes. Now, some of the electron microscopes that we looked at yesterday in our book, they made things look a million times bigger than they really are. I mean, I can't even wrap my head around that. I mean, what they use, instead of using light, they use a beam of electrons to produce a magnified image. And I'm not going to get into that any more than just saying that because it's very, very complicated. Okay, so as far as microscopes are concerned, you need to know about magnification and making something look bigger by using lenses. Obviously, the more lenses you have together, a compound microscope situation is going to make something look bigger than just having one lens alone. And then you want it to look sharp too. You want it to be clear, and that is resolution. So please make sure that you write a summary, and thank you so much. Is it still recording?